that Errol Akçay and I have been working on. And the model is stimulated by the fact that Kimberly LaPierre, who's a postdoc in my lab, is exploring the question of what the effect of enhanced carbon dioxide or increased nitrogen might have on the rhizobial community of 4 legume species that are involved in this biocon experiment, which is a long-term free air carbon enrichment experiment happening at the Cedar Creek Biological Lab. And as Jen Lau and Katie Heath have so elegantly illustrated, these long-term ecosystem studies have turned out to be really valuable long-term evolutionary experiments for studying microbial evolution. So just to bring everybody up to speed on the system, so legumes are plants. They enter into a symbiosis in which free-living rhizobia that live in the soil enter host plant roots. The plant produces a nodule, and the nodulated rhizobia then fix atmospheric nitrogen from the atmosphere, and of course plants are fixing carbon dioxide through photosynthesis, and then the legumes and the rhizobia trade these 2 resources, the carbon and the nitrogen, as you can see here. So because legumes and rhizobia are trading carbon and nitrogen in this mutualism, that means that those 2 resources, the effects of environmental variation in those 2 resources are going to be quite important for this interaction. Another thing to think about this system is, unlike the system that we heard about at the beginning of this session several hours ago, this is a system where there's horizontal transmission of the symbionts and the hosts. So we have a symbiotic life stage and an aposymbiotic life stage. The seeds are dispersed independently, and they don't contain rhizobia because the rhizobia infections are localized to the root nodules. And then the rhizobia are dispersed separately from the plants when these nodules senesce, and the rhizobia disperse out into the soil. And then the mutualism needs to be reestablished in the next generation. So another aspect of this system that's important to know is that multiple rhizobia of very diverse genotypes can end up in different nodules and sometimes even within the same nodule. So the fact that there's this independent reproduction is really important. And then the other thing to know about this mutualism is that this dinitrogen molecule, the 2 atoms of nitrogen, are linked by 3 carbon bonds, which means that breaking that carbon bond in the fixation reaction is very expensive. And so we have a system where there are very costly benefits provided and independent reproduction, and that can create a fitness trade-off in which selection is favoring mutations that reduce investment in nitrogen fixation. So we have selection pushing for rhizobia to getting more carbon for a particular amount of nitrogen, and plants are pushing, there's selection pushing plants to get more nitrogen for less carbon. So that is, has been thought to be a problem that might destabilize the mutualism. And it's been proposed, as we heard a couple talks ago, that there are a number of different mechanisms that could help stabilize this mutualism. And one that's been receiving a lot of attention recently is this mechanism by which phenotypically plastic traits of plants respond to rhizobia traits, fixation traits, and either choose partners that are more beneficial and avoid partners that are less beneficial. And then because this system is leaky, there's also the possibility that the plant might respond to the amount of nitrogen that it's getting from nodules and allocate resources to nodules with more beneficial strains. And so if those 2 systems were operating, we might expect to find cheating bacteria in fewer and smaller nodules. And there have been a number of empirical tests of this prediction now, and some of them show that there's evidence for these kinds of processes happening, and then there are other experiments that have shown that those processes may not actually be happening in some plants. But one of the major assumptions that hasn't really been very well examined about this process is that it assumes that the plant has complete control over the amount of carbon that's going into a nodule. And there's a problem with that, which is that if hosts have complete control, then there's nothing to prevent the plant from extorting the rhizobia. And so to start to try to resolve that question, Errol Akçay, who's my collaborator, along with his thesis advisor, Joan Roughgarden, developed a model, a negotiation model, which assumes that there are biochemical negotiations within the nodule, between a plant and a single nodule. And this model predicts that it will lead to a particular rate of nitrogen export into the plant and an initial carbon, and then a carbon export rate, and that these two carbon export rates will maximize the product of the fitness of these two systems. And what this does is that it then sets up a prediction for a particular price of nitrogen fixation. Okay? So, but this is only a single nodule, and so Errol and I worked on a model several years ago in which we looked at negotiation between the plant and two different nodules. And we came up with the idea of two different traits involved in this negotiation. One trait is this trait we call beta, which is the kind of the price that the rhizobia in a nodule that the plant is negotiating with are demanding for their nitrogen. And if they don't get that price, we assume that those rhizobia go out on strike. Okay, so the plant is like an employer negotiating with employees. Now, as the rhizobia demand more carbon, then the fitness of the rhizobia increases and the fitness of the plant decreases. Okay, so rhizobia with high beta are less cooperative. But at the same time that this is happening, a second nodule is there, and the rhizobia in that second nodule could provide an outside option for the plant, so that while it's in this dealing with this strike, these could be strike breakers over here. And so we have another trait, which we call kappa, which is the degree to which these rhizobia will increase their nitrogen fixation rate if the plant diverts carbon from this nodule to this nodule. Okay, so we have these two cooperation traits, and one thing to know about these two cooperation traits is that they have to be negatively genetically covarying for there to be variation in the population in cooperation. So for the plant to be able to choose between more and less cooperative rhizobia, you have to have this negative genetic covariance between these two traits. Okay, so given those models, now we then very recently set out to try to ask this question about how will increasing CO2 affect this interaction, and in particular how it will affect rhizobian cooperation. So we already explored how increasing nitrogen availability would affect the interaction, and we predicted, and it's since been shown, that it reduces rhizobian cooperation. And so I sort of naively intuited that increasing CO2 should increase rhizobian cooperation. But it turns out that it's a little more complex than that. So the simulation model that Errol has run assumes this negative covariance between these two traits. It also assumes that the carbon input to the plant, which is this variable, increases with increasing atmospheric CO2. And then it assumes that the price of nitrogen fixation, in other words, the rates of carbon export to the nodule and nitrogen export to the plant, that those rates are negotiated. So what did we find out? So what we found out has been a little puzzling to us, and we're just still working through what it suggests. So what we found is that as you increase carbon availability to the plant, so you can think about this axis as low atmospheric CO2 and high atmospheric CO2, that we do get an increase in the ecologically stable strategy of cooperativeness among the rhizobia, but it does this really weird thing. It has this hiccup here. So we've been wondering what is going on there. So first I want to expand this axis to show you in more detail what's happening. So we have this continuous increase in cooperativeness of the rhizobia, and then all of a sudden it plummets back down again. Although it doesn't go down quite as low, and in fact this happens multiple times, and each time it comes down to a little bit higher level. So we've been puzzling about why this might be happening. So what we think is happening is that as atmospheric CO2 rises, the plant needs more nitrogen to maintain its tissue carbon-nitrogen ratio, okay? And so that means that because the plant needs more nitrogen, the rhizobia can drive a harder bargain, which drives up this strike, going out on strike parameter. So the rhizobia become less cooperative, but due to this parameter. But the plant also has more carbon with which to reward that second nodule while it's negotiating with the nodule that's out on strike. Whoops. And so, this other kind of cooperation that gets expressed in the second nodule is also rising. And so it seems that the level of CO2 availability or carbon availability in the plant influences the relative importance of these two parameters during this negotiation phase. And so, so what, whoops. Did I, okay, there we go. Okay, so remember, since these two parameters have to be negatively genetically covariant for there to be any variation of cooperation, I think that's an important piece of this puzzle. Okay, so at some levels of carbon availability, kappa is rising really fast and beta is falling really fast. So the negotiating rhizobia are getting a poorer and poorer price for fixed nitrogen. And those are the rhizobia in nodule 1. And then, and so we see this rapidly accelerating rise as this price war between the nodules accelerates. And eventually it gets to the point where the rhizobia no longer get enough carbon to fix nitrogen. And at this point, at this level of CO2 availability, the importance of this stubbornness or being willing to go out on strike increases, and it very rapidly drives up the price. And that's sort of where we are at this point. And so what I can say in summary is that my intuition was correct that increasing carbon availability to the plant does seem to lead to an evolutionary increase in cooperation among the rhizobia. But it turns out that if there is this kind of negotiation going on, in other words, if rhizobia have control as well as the fact that plants have control, then we're going to see changes in cooperation that aren't necessarily just going to be a continuous rise in cooperation in these rhizobium populations. And so I'm really fascinated to see what Kimberly LaPierre and I are going to find when we start looking at cooperation variation in these different plots in the Viacom experiment. So I'll take the questions. That's something that Jen and Katie have also found, is that the density of rhizobia really shifts. And one of the things we want to extend this model to do is to explore what effect it has on the number of the sort of ideal number of nodules for a plant, which will kind of get at that density dependence. Any question?
Could you speak a little bit more about the cost of rhizobia going on strike and just hang out in the soil?
Well, so yeah, so what is that trait, in other words?
Well, what does it mean for the fitness-wise to go on strike?
So what we're envisioning is that going out on strike is a really short-term thing. So it happens at a very short time scale. And then over the longer time scale of how a plant is, of what rhizobia are getting from the plant is happening at a much longer time scale. So we're assuming that going out on strike is a very short effect. Errol, do you want to add more to that?
Yeah. So
Errol's the person who did the model, so he's much more.
I mean, it's very sort of basically just a kind of a hand wave way of motivating that they come to the beta, and this beta and kappa traits are sort of just influencing the position of the negotiation traits. So the beta, you can think of it as something either either trying to manipulate the plant or trying to get the plant to
Yeah, so
increase the carbon supply to the.
yeah, so I'll intervene and just say that, I mean, it could be something like, for example, right, a lot of rhizobium genomes have plant hormone mimics in them. So it could be that rhizobia have traits that, this is just kind of it's a toy model to try and get at some of those mechanisms. We aren't necessarily saying that the interaction happens as a strike. It's more saying that the rhizobia has a way of manipulating the plant. It has some control over the interaction, over the decision about the price of nitrogen, which the pure partner choice model assumes that the rhizobia are completely passive players in the price, setting the price.
So assuming this quick rise across a small range is correct, do you think it's possible that any empirical data is going to have fine enough resolution to be able to detect?
Yeah, so that's a really important question. So he's asking, given how short a range there is there, that we get this quick rise and then it drops back down again, will we have any resolution to empirically see this result? I don't think that we'll be able to see this jumping, but as we've run this out a little longer, we do see that the price does go up, and it would be interesting to know whether the price eventually, whether it continues to go up or whether some longer term drop happens. So I think, and we also don't, we don't have any parameter values, real life parameter values to parameterize this model yet. We don't really know what level of change in carbon dioxide concentration, whether it's at this scale or whether the change in carbon dioxide concentration is going to be at a much larger scale. We just don't have, we've only very recently looked at this. But what's interesting is that it's not this continuous linear effect, that it's a lot more complex interaction when you allow the rhizobia to have some control